Hi everyone, my name is Kitty Cam and I'm a master's student from Sir Ocean Lab, supervised by Dr. Doug Wallace. Today I'm going to present my topics about net parent production and net community production in the Center Labrador Sea. Before I go into my project, I just want to bring up some definitions to clarify the difference between the net parent production and net community production. So net parent production by definition is a gross parent production, which is the organic carbons produced from the photosynthesis process by parent producer, minus the autotrophic respiration, which is the loss of organic carbon due to respiration by phytoplankton. And net community production is the net parent production minus the heterotrophic respiration, which is the loss of organic carbon due to the respiration by heterotrophs. Or you can also see it as the gross parent production minus the sum of autotrophic and heterotrophic respiration. So the motivation of my studies is to understand biological carbon pump in the Labrador Sea. Biological carbon pump is one of the main carbon exports to the deep ocean, and it is directly linked to the net parent production and net community production. However, the constraints on the net parent production and net community production are not really well understood, especially the mixed layer depth and seasonal cycles, but they are essential to understand the bloom development and maintenance in the Labrador Sea. In the recent years, we have seen increasing autonomous measurements, but we rarely see the comparison between those platforms. And also we have a lack of high resolution in situ data for model validation in the Labrador Sea. Ultimately, we want to understand the implication of the climate change to the efficiency of the biological carbon pump. So with the support of the Phytoplot project, which is short for Ventilation, Interaction, and Transport across the Labrador Sea, CCycler was deployed from 2016 to 2017 to measure daily profiles of surface and subsurface biogeochemical properties, including oxygen, PCO2 nitrate, chlorophyll concentrations, and other physical and biooptical properties in high resolutions. CCycler is composed of 4 sections. First, you have the communication flow on the sea surface to talk to the satellite. And then you have the instrument flow that equipped with 13 sensors to make daily profiles from the sea surface to a depth of 150 meters. So within this surface layers, we're interested to look into the air-sea exchange, biological productivity and exports, transport and mass export, and also the mixed layer depth. And below this instrument flow, we have the mechanical winch flow, which is responsible to operate the movement of the instrument flow. And also we have equipped acoustic Doppler current profilers, like oxygen, PCO2 sensors, and CDD at this depth as well. And at the bottom of this CCycler mooring, we have the lower mooring, which we also equip another current profilers and a CDT, which specifically measure dissolved oxygen. So my projects focus on the comparison of the MPP and the NCP. So my first goal is to estimate 2 sets of MPPs from the in situ and remotely sense biological parameters using a one-dimensional parent production models. I use those MPP estimate to compare with the NCP in terms of magnitude, seasonal cycle, and dependency on the controlling factors, especially the mixed layer depth. So noted that the NCP is estimated based on the chemical properties, which are the dissolved inorganic carbons, oxygen, and nitrate. So this is just a basic schematic workflow of how I'm gonna estimate the MPP. So first we have the chlorophyll data and then the irradiance data from the sensor measurement. So for chlorophyll data, we're looking for depth profiles that measure from the surface to the bottom of the euphotic zones, which is where the parent production happens. And for the light parameters, we have the surface direct irradiance with wavelength within the photosynthetic light spectrum, which is between 400 nanometers and 700 nanometers. and also we have the diffusive coefficient of the downward irradiance. So with both of these values, we can compute a irradiance profile to input into the prime production models. Moreover, we also have two biological coefficients. The first one is the alpha B, which is the initial slope of the photosynthetic irradiance curve, and also we have the Pmax, which is the maximum prime production rate. So with all these values to put input into the prime production 1D models, then we can calculate a depth resolve net prime productions. So before we can actually go into directly compare the parameters and use them to estimate the net production, we ran into a few problems. So the first one is the CCycler and the satellite are measuring different light parameters. So from the CCycler, we got a downward irradiance at a single channel at 490 nanometers, which is measured from below the water surface. But for the satellite, it gives us a daily average PAR that is measured from above the water surface. And secondly, both platforms have quite a different temporal and spatial resolution. For CCycler, it has a really high temporal and spatial resolutions at a single locations. But for satellite, it covers a really fast space. It has a really fast spatial coverage. But for the regional resolution and the temporal resolution, it could be quite coarse. So therefore, we need to convert those parameters into the same temporal and spatial scales before making direct comparison and the MPP estimations. So the conversion approach for the CCycler and the satellites are slightly different, which I'll briefly go through in the next two slides. So for CCycler, when we get the raw data of the chlorophyll concentration profile, we apply a non-photo chemical quenching corrections onto those profiles to recover the loss of fluorescent signals due to the strong light intensity. It usually happens during the daylight or when the day or days with very bright suns. So basically, quenching happens when the phytoplankton cells dissipate excess energy, and then it will reduce the release of the photon energy as fluorescence. So once we recover those lost signals, then we will have an unquenched chlorophyll concentration profile, and which we can use it into the prime production models. For the downward irradiance profile, we'll use the curve and the logarithmic relationship to extract the downward irradiance just below the sea surface, and also we calculate the diffusive coefficient of this downward irradiance for each profile. And then we'll extract a full photosynthetic spectrum of the downward irradiance from 400 nanometers to 700 nanometers of the wavelength. With this, then we'll get the full spectrum of the downward irradiance and the diffusive coefficient to create a irradiance light profile for calculating the net prime production of the CCycler. For the satellite data, the surface chlorophyll concentration is averaged from the nearest 100 pixels available within the 40-kilometer circumference of the CCycler locations. Then I extrapolate the chlorophyll profiles up to the euphoric depth. For the radiance profile, the daily mean average PAR from the satellite is converted to an hourly PAR using the solar altitude angles, which is the angle between the sunlight with respect to the ocean surface. And it is unique to location and time as well. Then I convert the PAR to the downward irradiance within the photosynthetic spectrums using these mathematical relationships. And my last step is to convert the downward radiance measured from above the water surface to below the surface. because not all incidents penetrate into the waters, and some might be reflected depending on the wavelength and the water surface conditions. So for each wavelength, they have their specific conversion coefficients to convert from above the surface to below surface. And this is the reason why I didn't directly convert the daily par from above the surface to below surface for these reasons. With all these values, the chlorophyll profiles and the radiance profile and the diffuse coefficients of the downward irradiance, then I put it into the parent production models to calculate the net parent production of the satellites. So just to recap what I have talked about in these presentations. So first, my project is to compare the net community production and net parent production estimate of the CCycler and the satellites in the Labrador Sea in terms of the magnitude, seasonal cycles, and the dependency on the control factor, especially the mixed layer depth. And in the future, we're planning to incorporate the SCP estimate from the biogeochemical flow as well to compare with the estimate from the CCycler and satellite as well. And the take-home message of this presentation is that the CCycler gives a long-term fixed location hive in high-frequency carbon-based estimate, and it is quite rare to have such a high-resolution data set to compare with the satellite data. And also in situ data is very important to serve as purpose for its validation for the regional models and remote sensing algorithm. So for all this is my reference from a presentation, and thank you, and I'm happy to take any questions.